Good morning and good afternoon. It is my absolute pleasure to moderate this session and to introduce our speaker today. My name is Kathleen Cox, and I am an Associate Vice President of Drug Disposition at Eli Lilly. Dave Coutant is our speaker for today. Dave graduated with his PhD from the University of Michigan in Ann Arbor with a degree in analytical chemistry. He's been employed at Eli Lilly for 24 years now, working in the drug disposition department. He's also regulatory affairs certified. Dave has worked on many drugs over his career, both small molecule and biologics. He has over 20 journal articles, including 3 on the topic that he's going to speak about today, disease-mediated drug interactions. Most recently, Dave chaired the IQ leadership group commenting group on the FDA guidance on therapeutic drug interactions. And in 2021, he formed the IQ-TAL gene therapeutic protein working group. So without further delay, it is my pleasure to introduce Dave Coutant.
Michael J. Fox once said that medical science has proven time and again that when resources are provided, great progress can be made in the treatment, cure, and prevention of disease. Now, one of the key aspects of effective drug treatment and patient care is understanding drug interactions. My pleasure today to be able to talk to you about therapeutic protein drug interactions. This webinar is a sister companion to a white paper that was put out by the IQ. That white paper was a collaboration among 13 different companies, and I have many experts to thank for their invaluable contributions to that white paper. They're listed at the bottom, but in short, many thanks are owed to David, Upendra, Antoine, Christine, Jobao, Lena, Hatal, Hilmar, Wao Ying, Kenichi, Yang, and in particular, Shannon. Now, the IQ, or International Consortium for Innovation and Quality in Pharmaceutical Development, as you know, is a non-for-profit, technically based consortium of over 36 companies. The mission statement is displayed here. In particular, this webinar was brought to you on behalf of the Translational and Agri Sciences Leadership Group, the TAL. We also did work in collaboration with the Clin Pharm Group. You can see both of those in kind of the upper right of this picture. Now today, we're going to be talking about several different types of therapeutic protein drug interactions. The primary focus is going to be on cytokine-mediated drug interactions, right? And that falls into 3 subcategories. Cytokine modulation caused by indirect disease drug interactions, which can be caused by moderate to severe inflammation, injury, or infection. Direct dosing of pro-inflammatory cytokines, or third, indirect drug interactions due to dosing of therapeutic proteins that are cytokine antagonists. The other areas of drug interactions we'll look at will be drug interactions related to antibody drug conjugates, or ADCs. And the third kind of catch-all category, those related to, not related to cytokines, not related to ADCs, which we'll talk about, but involve TMDD, FcRn, antibody, antidrug antibodies, etc. Once we get through those areas of therapeutic protein drug interactions, we're going to then delve into what are some of the practical considerations for evaluating these types of drug interactions. We'll talk a bit about inflammatory markers. So what is possible? What are the limitations of using inflammatory markers as kind of a assessment to help categorize the overall level of disease drug interaction risk? And then finally, conclusions, recommendations, future directions. Introduction. Since 2008, sponsors in pharma and biotech have completed numerous therapeutic protein drug interaction, TPDI studies, for various cytokine antagonists in disease states from psoriasis and atopic derm all the way to Crohn's disease and rheumatoid arthritis. The cause and the risk amounts, the level of risk in these different diseases and for these different types of cytokines is lack clarity. Not so long ago, in August 2020, the FDA put out a draft guidance on therapeutic protein drug interactions. As a response to that, the IQ formed a commenting group, and that had members from both the TAUG as well as the Clin Pharm leadership group. They combined to issue a set of comments. Those are publicly available. Those comments were the basis for the white paper and this webinar that came out of it. A key point, we'll start now and you'll hear several times today, is that for cytokine modulation, it is disease state and disease severity that are the primary determinants of your risk level, your magnitude of drug interaction, more so than the type of cytokine antagonist. So basic assumptions as we move forward, we're going to treat the magnitude of these drug interactions just as for large molecules, just as we would for small molecules, right? So the change in AUC of a sensitive drug substrate, if that change in AUC is less than 1.25, we'll declare that negligible, right? A weak interaction is 1.25 to 2, etc. Focus of discussion. We're going to be talking about therapeutic protein DIs of clinical concern, right? Just broadly speaking, TPDIs, where the systemic exposure of the victim drug is changed at least twofold. There are exceptions to the rule. Warfarin, for instance, has a narrow therapeutic index. It's a sensitive substrate. Changes in its AUC less than twofold can be clinically concerning. But for the most part, we have enough of a therapeutic window that we're going to be focusing today on those disease states, those causes of cytokine modulation that create at least a twofold change in AUC. We're going to talk about representative examples. We will not be comprehensive. We don't have time. Outside of the scope of today's talk are going to be a number of non-traditional small molecules. So we'll treat siRNAs, ASOs, acylated peptides, as small molecules, not discuss them further today. Cytokine fusion proteins, cell or gene therapy, those are very exciting targets, but they have not been in the clinic long enough to have a number of new novel therapeutic protein drug interactions. So there's off the table. And then we will again try and focus more on what is clinically observed, more so than theoretical drug interaction risk. So we'll move into our primary topic for today. The body's reaction to inflammation consists of a complex response to autoimmune disease, infection, tissue damage, and tumors, right? So as a result of that, the acute phase response includes both local and systemic reactions. As a result, we'll take moderate to severe inflammation. That can cause, right, an increase in the activity of nitric oxide synthase too, which can result in direct decrease. It can directly inhibit the cytochrome P450 activity. That's a minor pathway. What we're going to focus on is the pathway where that moderate to severe inflammation causes an increase in several pro-inflammatory cytokines. The ones most studied are listed here on the slide. Those increased pro-inflammatory cytokines in turn then hit a complex interplay of transcription factors, which then ends up resulting in the decreased expression of one or more cytokines P450, possibly some drug transporters, and that results in decreased CYP-mediated drug clearance. Here is a case example based on clinical data. So on the leftmost panel, we have simvastatin being given daily. You can see the rise and fall of simvastatin. So you have a healthy patient in the leftmost panel, healthy, other than the fact they have high cholesterol. Now from the leftmost panel, the middle panel, unfortunately, this patient also develops rheumatoid arthritis. What is the outcome of that? Well, as an outcome of that moderate to severe inflammation in this particular group of patients, right, your pro-inflammatory cytokines go up, a number of cytokines increase. Turns out there's literature data to say that IL-6, interleukin-6 is the one that increases most. Multiple CYPs, some transporters are affected. It looks like, again, from literature data, that predominantly, in this case for RA, CYP3A4, possibly, let's say probably PGP, they are decreased in their expression. As a result, the exposure of simvastatin at the Cmax increases on the order of about threefold. Now, as we move from the middle panel to the rightmost panel, that patient is treated. Right? In this case, it was with tocilizumab, Actemra. This is a drug interaction study, so the duration was not long, right? So they could not be fully treated, but even after very short treatment, what you see is normalization of CYP levels, PGP levels, and your simvastatin levels start to normalize. This is an example of what's going on in rheumatoid arthritis, but this is the similar type of thing that we can expect in other disease states. So what other disease states are of interest? It's a very busy slide. I want to remind everyone that this seminar is a companion to the white paper, and so all of the major tables and figures, they're all in the white paper. You can see them there for more data. This is the table, not of every cytokine modulation that leads to a disease-drug interaction, but the ones that are of most concern, greater than twofold change, right? So as you go down the leftmost column, infection, such as influenza, HIV infection, further down the table, infections due to COVID-19, various other severe medical conditions, critically ill, bone marrow transplant, moderate to severe inflammation, sepsis, RA, Crohn's disease, non-alcoholic steatohepatitis. All of these have shown at least twofold changes in these sensitive substrates. These are the conditions right now that are of greater concern. In contrast to a combing of the literature and what are the disease states and conditions are of greatest concern, what are the ones that have been studied since 2000? So here's a summary, right, gathered from clinicaltrials.gov of the completed therapeutic protein drug interaction studies that have been conducted. Right? And you can see the different modes of action. This slide, however, has been grouped primarily by the second column, that of the disease type of patient population. And the takeaway for this slide is that in these studies, atopic derm, multiple sclerosis, osteoporosis, multiple studies in psoriasis, no clinically meaningful drug interaction was observed in any of these cases, right? They may have an inflammatory component, but it was not enough to elicit a decrease in CYP activity that was noticeable. Only in the 3 instances in rheumatoid arthritis did we see a drug interaction. I also did want to mention in passing, studies are ongoing in a mixed population of UC and Crohn's patients from more than one company. Those have not read out publicly, so they're not included in the slide. So as a more comprehensive slide, we move on to this figure, which is to summarize what we know at this time. It's a living diagram. We're going to have to update it as we get more information. In the upper left are all the disease states that were shown 2 slides ago in that table, the disease states of greatest concern. We see moderate to strong change in the AUC of a sensitive substrate, right? Influenza B all the way down to MASH. Below that, in the lower left, this is where we see we've grouped possible disease states of concern, right? We don't have as much data. We don't have 2 or more clinical DDI trials that have shown risk. We have inflammatory markers. Perhaps we have some effects in transporters that suggest maybe as we get more data, we might see a moderate level of drug interaction. In the upper right, what is established as weak interactions, right, from cancer, hepatitis B, all the way down to NAFLD. Possible weak interactions on the middle right, based mostly on inflammatory markers, right? So inconclusive, there might be no interaction, there might be a weak interaction, we need more data. And the one we feel pretty good about due to numerous studies being conducted across multiple sponsors, is that psoriasis, again and again, is not shown disease-drug interaction. All right. What else do we want to say about therapeutic protein-drug interactions due to cytokine modulation? So we've talked about, we listed the table of all the different cytokine antagonists that have been studied. We've talked about the disease states. The third subcategory is that of dosing of cytokines themselves, right? So there have been instances such as dosing IL-2 as a therapeutic to cancer patients that have shown that direct administration of a pro-inflammatory cytokine can decrease CYP content. Now, transporters, conjugative enzymes, they are only a minor focus of this talk in the white paper. They're much less studied. They're also not, it's much harder to find, if not impossible to find, a very sensitive substrate. And so interpretation of drug interaction effects involving transporters and UGTs is a little bit harder. There's preclinical data, non-clinical data with both of these that show that disease-drug interaction, cytokine modulation can occur with transporters and conjugative enzymes, but that is less of a focus, harder to understand. So to group all of these together, right, we have under the umbrella of cytokine-related mechanisms, we've got the three categories that we've talked about. But as we wrap up cytokine modulation, I want to reiterate the take-home messages. Moderate to severe inflammation, injury, infection can decrease CYP expression, and successful treatment can normalize or raise CYP expression. This occurs in select circumstances. Most of the inflammatory disease states are going to have insufficient systemic inflammation to cause concern. The type of disease, the severity of disease are the key factors in assessing therapeutic protein-drug interaction risk and predicting the magnitude of that drug interaction. Now, we move away from cytokine modulation. The next category would be that of ADCs, antibody-drug conjugates. Very exciting field of research. It's not surprising then in the 2018 timeframe, an IQ work group was formed to look at ADCs. They looked at the biological challenges of measuring them, as well as also drug interactions of ADCs. That white paper is now publicly available at this site. I suggest everyone that's read it. It's very comprehensive. As such, we will not spend a lot of time recapitulating what all of the conclusions from that paper. That paper did point out, of course, that drug interaction potential can arise from ADC either from the antibody portion or from the small molecule drug payload portion of the ADC. The IQ white paper did not comment on the theoretical risk of what if that ADC is treating an autoimmune state or cancer state where there's moderate to severe inflammation. Could you have indirect cytokine modulation and drug interaction in that regard? So we put all that together, their findings, our findings from our group, those are summarized at the bottom of the slide, right? So overall, first two bullets are their conclusion. There's a negligible risk for an ADC payload to be the perpetrator of a drug interaction. That's because simply that drug payload, since it's being targeted to the site of action, its systemic concentration is so small that the concentrations are extremely rarely going to be high enough to elicit inhibition of a CYP or a transporter. There's a possible risk for that small molecule payload to be a victim of a drug interaction depending on what it is a substrate for. Our group, we postulate that there's a theoretical risk for immunomodulatory ADCs to cause indirect drug interactions. Personally, based on the figure, the photo in the upper right, my conclusion is that all ADCs are nearsighted. So the next category, the overall category, the catch-all, what about the 4 subcategories that aren't related to ADCs or cytokine modulation? So the first one is, what if you're dosing of therapeutic protein with a concomitant medicine that impacts TMDD, impacts target-mediated drug disposition. Right? So when clearance of a victim therapeutic protein is dependent on TMDD, and the con-med changes the concentration of the target ligand, or it competes for the same ligand, that can result in this type of drug interaction, right? Because you're looking, if your compound, right, the one that you're developing, that therapeutic protein is something that is dependent on TMDD, you could look at whether or not it's a victim of a drug interaction using population PK, population pharmacokinetics in phase 2 and phase 3. You wouldn't need necessarily a standalone study. The second subcategory would be that of a therapeutic protein causing a physiological change. For instance, incretin mimetics have been shown that they delay gastric emptying, right? And so that can be investigated clinically in healthy subjects. You can look at the change in gastric emptying in that way, and you can use PBPK as a companion to that. Now, the other category would be that of FcRn. So extremely high doses of immunoglobulins, IgGs, have overwhelmed the capacity of the neonatal Fc receptor in some cases and altered IgG antibody clearance. So if you have a con-med or a disease that could alter the function of FcRn, you might therefore have a drug interaction in this regard. In particular, if you have a drug that is an IgG or is dependent on FcRn for its long half-life, you can, with certain other con-meds, you can alter the clearance of that therapeutic protein. The fourth area would be that of immunogenicity-dependent drug interactions, right? Antidrug antibodies. So if you have, let's say, take something like infliximab. So infliximab has, with some variability, a range of antidrug antibodies. So there's a range of immunogenicity that we see in the clinic. If you co-administer with methotrexate, that can reduce that has been shown to reduce the overall amount of ADAs. And as a result, you're going to change your systemic exposure, right? Immunogenicity was reduced with methotrexate, and that resulted in an apparent reduction in clearance of infliximab. That is the grouping of all the different types of therapeutic protein drug interactions. So we're going to move into some practical considerations for their risk assessment. In vitro, in vivo models of therapeutic protein drug interactions. How useful are they? Well, to answer that question, we look at what has been done in the past. A number of the cases of in vitro investigations of cytokine modulation have taken a single cytokine and spiked it into human hepatocytes. That has some utility. It can help tell the scientist what are the cytokines for P450 or transporters that are being affected by that particular cytokine. The limitation, the caveat to this is that it doesn't look at the complex interplay of all the different cytokines and all the different transcription factors. So for instance, as a case example, interleukin 2 does not have receptors in human hepatocytes. Therefore, it has been previously postulated that if you are dosing a IL-2 cytokine antagonist, you would not elicit a indirect disease drug interaction. However, in contrast to that, right, direct dosing of IL-2 in cancer patients resulted in an overall total CYP expression decrease of about a third. It's postulated that due to that complex interplay of cytokines, although you might not be affecting IL-2 directly, everything that's going on can still result in this type of indirect disease drug interaction. So in vitro models have to be taken with a grain of salt, so to speak. Non-clinical in vivo models, right? Those also have some utility, but have to be interpreted cautiously. For instance, the diabetic OB rodent model is often used as a diabetes model. And in those rodents, you can see if you administer a very sensitive CYP substrate, several of them will have much decreased CYP activity, increased concentrations of your small molecule drug compared to that in normal rodent. However, that same very large decrease in CYP content is not seen clinically, right? In type 2 diabetics in the clinic, we've only seen negligible to weak changes in CYP expression. So both in vitro and the non-clinical in vivo models, they do have utility in helping us understand things from a mechanistic point of view, but from a predictive point of view, particularly with regards to cytokine modulation, they're not there yet. They're just not predictive tools in that regard. PBPK modeling. So in terms of PBPK modeling for cytokine modulation, what we have here is a summary of several instances where the authors, a team of authors, have gone and tried to use different means to try and model this type of cytokine modulation. For instance, Simon used a top-down approach in C-reactive protein to try and model the changes in IL-6 and the subsequent changes in CYP expression. Overall, these models do a fairly good job retrospectively, but from a prospective manner, they're not there yet. It's very difficult to fully understand the time course of cytokine modulation, which cytokines are being affected. We don't have all of the information that we need in terms of Emax, various other parameters to be able to dial this down completely, dial it in completely to what we would want. Now, PBPK modeling does have its strengths. For instance, one of the things that we mentioned before was that incretin mimetics delay gastric emptying. So exenatide, which is a synthetic analog of a particular peptide found in the saliva of a Gila monster, which affects GLP-1. That was one of the first to show the delay in gastric emptying. And so Morse and others most recently went in, and it's a published article, and this showed, for instance, that if you do a drug interaction study and you co-administer a incretin mimetic with something like atorvastatin, you can see the delay in gastric emptying, and then you can, from that information, use that in combination with PBPK modeling to help predict what would happen in other compounds whose overall disposition is dependent on gastric emptying. So PBPK modeling, it has some limitations in terms of predictivity of cytokine modulation, but it does have some good utility in some other regards for therapeutic protein drug interactions. Clinical design considerations. So if we have some limitations, right, we have some considerations for what we can learn in vitro, in vivo, PBPK, what about our clinical investigations? Well, as the audience knows, most of our therapeutic proteins are going to have very long half-lives. So instead of a small molecule doing a crossover design, we may have to, due to the long half-life of the therapeutic protein, do more of a parallel design. Increase N, increase cost, increase time of the study to be able to do a drug interaction study for therapeutic proteins compared to a small molecule. Now, if we clinically investigate a TPDI, what is the patient population, right? Is it healthy volunteers, is it patients? Depends on the type of drug interaction that you're investigating. For cytokine modulation, for disease drug interaction, it is advised that you study it in the patient population, and particularly the most sensitive patient population, right? So if ultimately a sponsor is going to register a drug for both psoriasis and rheumatoid arthritis, doing a disease drug interaction study, right, looking for cytokine modulation in healthy subjects or psoriasis patients will not yield any measurable drug interaction. However, doing it in the RA patients, you may see a drug interaction depending on the severity of RA in that particular population. Now, if the drug interaction you're looking at isn't related to cytokine modulation, let's say that your compound is dependent, has its clearance dependent on TMDD, then you may have the advantage of not having to do a standalone trial, but instead use population pharmacokinetics in phase 2 and phase 3 to determine whether or not your therapeutic protein is a victim of a DDI. Labeling. One thing to consider is that there may be instances that clinical investigation of a drug interaction is going to be very difficult. So in particular, we'll look at Crohn's disease and cancer patients that have very advanced or metastatic cancer. Crohn's disease is very sporadic in terms of its onset. Patient population is very variable and very diverse. Conduct of a therapeutic protein drug interaction study in Crohn's patients can take years just to enroll. And then at the end of the study, those results may be confounded due to all that variability. Cancer patients, extremely severe cancer patients, you may have ethical issues in conduct as well as, understandably, a lot of constraints in just the simple conduct of the study. In instances such as those, sponsors may want to consider whether or not just to propose simple label language in place in lieu of a clinical therapeutic protein drug interaction study. Right? Labeling could suggest caution when dosing sensitive substrates or words to that effect. Now, I would be remiss if I also did not throw out that, again, if I didn't reiterate that disease drug interactions occur in select circumstances. The majority of diseases, although they may have a systemic inflammatory component, that systemic inflammation is so low that you're not going to cause any type of meaningful drug interaction. So how do we characterize the compounds that are of negligible, weak, moderate, severe risk? We would propose that these inflammatory markers, although not perfect, may be of use. Use of 2 or more of these inflammatory markers may help you bin or categorize a particular patient population as having negligible risk, weak risk, etc. C-reactive protein, albumin, neutrophils and lymphocytes with the neutrophil-lymphocyte ratio, AAG, cytokines such as those listed here, have all shown fair productivity in terms of changes in those particular inflammatory cytokines corresponding in broad strokes with the level of inflammation and thus the level of changes in CYP content. Now the caveat, I want to be clear about this, it is understood these biomarkers are highly variable, right? Even within a patient, from day to day, over the course of a day, and from day to day, there's going to be a lot of variability. We are certainly nowhere near the point where we can pick a C-reactive protein level in a patient or an albumin level in a patient and precisely dose adjust. What we are proposing is that these inflammatory markers used in tandem, that the mean levels for a particular population may help you bin approximately the level of concern. Now, in the future, it may be that there are some advances that might help. So for instance, liquid biopsies, right? Looking at the mRNA of certain CYPs in plasma isolated exosomes, in the paper shown here, has recently shown pretty good concordance between the levels in those exosomes compared to the overall CYP activity and what type of drug interaction you might have. That needs a lot of work. Right now, just a single draw for a liquid biopsy is going to be very expensive. Right, so hope for the future perhaps. Here is an example of two of the inflammatory markers. And so we've known for decades that as you increase inflammation, albumin levels decrease and AAG levels increase. And you can see on this slide, as you move from the healthy participants on the left to the more moderate to severe inflammation on the right, you can see these trends. You also see a lot of variability in the data and a lot of overlap, right? So these are not perfect. Interleukin 6 is the cytokine that has been looked at most often, right? So we've talked about interleukin 1, interleukin 2, IL-6, TNF alpha, interferon gamma. Those are the cytokine, pro-inflammatory cytokines that have been looked at most often in terms of affecting CYP activity. Those have not been measured in every disease state, right? IL-6 is most often measured, if at all. You can see here how going from the bottom of the slide to the top, that as you go from healthy subjects all the way up to much more severe inflammation, you can see how IL-6 increases. You can also see the error bars. I also want to point out, you go 2 or 3 lines up from the bottom, congestive heart failure, mild versus severe, you can see that disease severity plays a difference. Or as you go 5 or 6 lines from the top and you look at Crohn's disease, active versus inactive, again, you can see mean differences in these data sets. So it's not only disease type, but disease severity that can impact markers such as IL-6. In particular for rheumatoid arthritis, we have captured here, I don't want to say this is a cliff and anything beyond it is concern and anything above it is not a concern, but some general guidelines for rheumatoid arthritis is where might you start to see concern. So for these pro-inflammatory markers, IL-6, CRP, albumin, AAG, in the middle column you can see the upper limit of the normal range. So if you have your systemic concentrations of these inflammatory markers are next to close to the normal range, you would expect little to no inflammation, little to no change on CYP activity. Far right column, right? As you look at, for instance, a rheumatoid arthritis population, you're trying to assess, okay, what is the level of inflammation in this particular population? So take it across all the individuals, get a mean, compare that mean for C-reactive protein. If it's above 50 milligrams per liter, if in that population albumin is close to 35 grams per liter or below, I personally would be concerned that you're in the range of at least a twofold drug interaction. If those markers for CRP are closer to 20, let's say IL-6 and CRP are closer to 15, and albumin is closer to 38, you probably have a mild population with mild RA, and you might only see a weak drug interaction. In conclusion, therapeutic proteins overall have a low risk for eliciting meaningful drug interactions, right? Therapeutic proteins of clinical concern can occur in select instances, however. We need to understand what those instances are. Drug interactions can involve a variety of means for therapeutic proteins, including cytokine modulation, TMDD, FcRn, ADAs, physiological changes. In the arena of cytokine modulation, right, we know that it can occur by direct dosing of the cytokines, by cytokine modulators being administered, or by indirect disease drug interactions caused by injury, infection, inflammation. Type of disease and the severity of the inflammation are primary drivers for the magnitude of the disease drug interaction involving cytokine modulation. While there are limitations use of some of these clinical inflammatory markers very broadly may help us categorize which disease states we do not need to further investigate, right? It may help us categorize negligible and weak and moderate drug interaction risk for these different inflammatory disease states. And as an outcome of that, perhaps in the future, we can use those inflammatory markers to say, okay, this disease state doesn't even merit investigation. Certainly, in terms of clinical investigation, psoriasis is something that we've studied several times. And as a recommendation out of that is, owing to the fact there have been 8 therapeutic protein drug interactions studies conducted in psoriasis patients across 6 companies, we have not seen any meaningful drug interactions. We recommend that drug interaction studies, disease drug interaction studies involving psoriasis patients just don't need to be done. We've already talked about the second point. The third point, we'll recommend to everyone that if you understand the type of therapeutic protein drug interaction that you're looking at, that will help you understand what type of characterization you need to do. Does it need to be a clinical study? Can it be pop PK? Does it need to be in patients versus healthy participants? Our last recommendation was that looking at the 2020 FDA guidance, we thought it was very fine. We did suggest, as in the blue type here on the upper left, a few changes basically to show that there are what we consider to be the 3 subcategories of cytokine modulation. As we move forward, future directions. Well, we would hope to gather more information about indirect disease drug interactions, right? In what regard? Well, in one case, we've only measured cytokines in a few studies. We haven't looked at TNF alpha, IL-2. There's a lot of cytokine levels, a lot of inflammatory markers that we don't know what their levels are in ulcerative colitis and Sjogren's syndrome and lupus and many other disease states. So we'd like to gather more markers, more data. There are other things, right? We need more information about transporters and conjugated enzymes. There's also questions, for instance, as to does the decreased CYP expression, does it parallel both in the liver and the GI system? Data is inconclusive. These are some of the future directions. By gathering that data, looking at it, we hope that we can understand what are the disease states of concern and which ones are not. Finally, PBPK tools. It's always an aspirational goal that we can get to the point that we have a virtual human, and that will help everybody, sponsors, HCPs, patients, as we can get, we can characterize drug interactions, we can predict them with greater efficacy, with greater efficiency and speed. So overall, there are many factors that need to be considered in the assessment of drug interactions. So what is your type of drug interaction? Right? Is it involved TMDD? Is it ADAs? Is it cytokine modulation? What is your patient population? Overall, a weighted approach examining multiple inputs, right, is the best way to interrogate therapeutic protein drug interactions. And then from that, decide what is the best way to assess it. I want to thank all of the members of the IQ working group that helped create the white paper. They were on the first slide and listed all here again. I want to thank Kathy Cox as our sponsor, as well as Raymond Evers of Janssen, Steve Hall of Lilly for some very extremely helpful and detailed feedback on early drafts of that white paper, Swapan Chowdhury for early inputs. I want to thank all the members of the 16 plus companies that reviewed early versions of that white paper. And I want to thank everyone online for their time in listening to this talk. I will now pause to be happy to take any questions from our moderator today.
Excellent presentation, Dave. Thank you very much. It's clear that you're very passionate about this topic, and thanks to your team as well. For folks who have questions, go ahead and enter them into the question category. There's one already in there. Thank you, David, for the great tutorial. In relation to phase 2 and 3 studies, did the team look at how intensive or at what time points in relation to drug dosing the cytokine should be for the info to be used later in the POP PK?
Excellent question. So to reiterate, if you are looking at phase 2 or phase 3 dosing, and you're trying to assess cytokine modulation, you know, how is that all assessed? What time points? Well, first off, a lot of therapeutic protein drug interactions up to this point have really focused on kind of doing a dedicated therapeutic protein drug interaction study. This is a bit of a switch to looking at trying to assess through use of inflammatory markers in phase 2. In phase 2, commonly, what we're going to do is, some cases you might do a full PK profile, but more often, due to the nature of the study, is you're going to be using population pharmacokinetics, right? And so it would be difficult to push on, take PK time points at these certain days or these certain time points to answer your questions about a potential drug interaction. We have to use the data that we have. But you raise an excellent point, which is there is a time element to all of these changes in CYP expression, right? So cytochromes P450, their synthesis degradation rate is on the order of, let's say, for instance, for CYP3A4 is on the order of 36 hours. So that about a day and a half. So if you have a spike, let's say from a fever, or you have rheumatoid arthritis, even though your body may feel acute phase response right there, the change in CYP expression to hit a new steady state is going to take 5 half-lives. It's going to take about a week. So also, for instance, albumin, it's got a much longer synthesis degradation rate. So that's closer to 2 weeks. So that is to say that if you looked at albumin levels or your CYP levels a day after your dose of your compound, that would be insufficient time to see a change. You would want to say, okay, maybe on day 14 of the study, right, as you do pop PK every so often, you can get kind of a baseline read before you start giving your drug, right? That's going to give you the baseline level of inflammation in the disease state. And then later, once you've gotten to a new steady state after disease treatment, you can see how much it's gone down. I think right now, the greatest amount of data is what are the levels of these inflammatory markers at baseline before you give the drug. That's the level of information I'm interested to get right now.
Good, thank you. The questions are popping in now. So I have another one for you. Is there an effort or database that has collected data on prototype substrate drugs in specific inflammatory diseases to help us characterize the enzyme? Just popped off. Sorry. Let me start again on that one. Is there an effort or database that has collected data on prototype substrate drugs in specific inflammatory diseases to help us characterize the enzyme transporter activity in those patients?
Transporter activity in those patients. The short answer is no, unfortunately. A lot of this webinar is showing, I think, illustrative of a shift kind of in thinking. Part of the FDA guidance was also a shift that it's more disease state than it is cytokine modulator. And so as such, the idea that it's a disease state, and then in a particular disease state, what are all the inflammatory markers and what are all the changes in CYPs and what are all the changes in transporters? that just hasn't been done yet. One example is Emily Schwenger and others in AstraZeneca put a paper out a few years ago on cancer patients where they took the standard SimCip PBPK model for cancer patient and then combed through all the data and said, okay, based on this, what changes do we need to make in CYP expression for this CYP, this CYP, this CYP, this transporter? I'd like to see that, gather the data so we can do that in more diseases. Good question. It's not quite there yet.
Next question is from Camelia Benya, GSK. Do you have any comment on how to assess the potential for DDI for small molecule immune modulators that will induce cytokine release like IL-6 due to target activation, but also are substrates for 304?
Okay, excellent question. So first, kudos to you. Some people have resisted the idea that disease-drug interaction can affect small molecules as well as large molecules. Certainly, I've seen several instances with internal data that show that disease-drug interaction effects due to moderate to severe inflammation can also affect small molecules. How would I characterize it? Well, first thing I would do is look at the indications that you are pursuing and compare that to those disease states that are already, that just put out in this white paper. So if you are looking at atopic dermatitis, psoriasis, I would say that you're in the clear. That disease is not going to affect your drug. Other things that have an inflammatory component, osteoarthritis, Alzheimer's, etc., diabetes, probably not a concern. If you don't have data on your particular indication, then that's where in phase 2, right, look at the PK of your compound in phase 2 in patients compared to in healthy subjects, see if it changes. Now, your compound is it a sensitive substrate in and of itself? Right, if your compound is cleared in multiple avenues, then a change in CYP expression isn't going to affect the overall clearance. If it's a sensitive substrate of CYP3A, then you would expect that indeed your small molecule drug would have a different exposure in patients versus healthy subjects. So first I would look at, try and look around and see, am I in a disease state? Do I have a compound who's, am I in a disease state of concern? Do I have a compound that is a sensitive substrate? If that Venn diagram doesn't create risk, then you're done. If it does create some risk, then you can characterize a bit further by inflammatory markers, comparing the two, some simple PBPK models. No one yet, there's no regulatory, I will say there's no regulatory agency that has requested any small molecule drug to do a disease-drug interaction study.
Next question. For rare disease categories, what if not much info is known on the mechanism of action of the diseases? How should the team approach assessing the necessity for a drug-drug interaction, or how to justify a waiver?
So very good question. Rare diseases are concerning. Probably what you would do is look at the data that you have. Is there anything that you do know about your compound? So as you approach submission, even for a rare disease, you would probably know, are there, what is the chances your compound is cleared by TMDD? I don't think a lot of incretin mimetics are going for rare diseases. Drug-drug interaction based on immunogenicity is not a big concern. TMDD, you'll probably know. FcRn, you would probably know. Cytokine modulation depends on the disease state. You would look at, I would say, your therapeutic window, right? And consider that most of the therapeutic protein drug interactions are going to be quite small in magnitude compared to your therapeutic window. If your therapeutic window is extremely small, you might be in an autoimmune disease state that's high inflammation. Maybe in that case, you would put in some of that label language like I suggested for Crohn's and cancer, where you just say, sensitive substrates of CYPs might be dosed cautiously, that type of thing.
If only the severe inflammation leads to CYP suppression, which cytokine and what levels of those cytokines is considered a severe inflammation?
Great question. I would have loved to have a table answering that exact question in our white paper and in the seminar. The data just doesn't exist, and that's why it's one of the future directions. I'd like to know more about, we just don't gather as sponsors in the industry. You don't gather a battery of 7 inflammatory markers in phase 2. We just haven't to this point. If you can get 2 or 3 in there, great. You know, what are they at baseline? Compare that to upper limits, the limits of the upper range. Are they close? At the moment, as you go back to that one slide, really the only table that we felt comfortable putting that all together for was for rheumatoid arthritis, right? And so in that case, as you look at, for instance, IL-6 and C-reactive protein, from if you have a mean population of, say, 5 picograms per mil for IL-6 and 5 milligrams per liter for CRP, up to 20, probably weak interaction, right? As you get to CRP and IL-6 greater than 50 picograms per mil as a mean for the population, that's where I'd be very concerned. Do some literature searches that can help. Otherwise, I think we just have to collect more data.
Have you heard of an FcRn therapeutic protein DDI that could be due to the pH increase in the endosome caused by co-administered drug, for example, weekly basic drugs, that may alter the binding of the therapeutic protein to FcRn and thus affect its clearance?
So 2 things. So that's a very good question. I know we're near the top of the hour. Also, admittedly, I am more of an expert on cytokine modulation than I am FcRn. So I'm going to ask in this case, I don't know off the top of my head any such example. However, what I will pledge to do is if we can capture that question, I will, I'll do some quick research and get back to you.
Okay, good. And we are at the top of the hour. There are several more questions in here. And what we will do is we will make sure that Dave has the questions and we'll provide the answers offline through the IQ secretary. Also, there was a question in here about making the slides available. The slides will be made available in addition to this recording. So I would like to thank Dave and his team for a very thorough discussion, very helpful, lots of interest. And I would like to thank everyone online for participating. Thanks for your time.